Best regards to everyone. Our project is Annotation of transposable elements in coffee roast. This was developed by Mariana, Sofía, Candamil, Paula, Andrea, Jaimes, Reinel, Tabares-Soto, Cristian, Felipe, Jiménez, Simón, Orozco-Arias, and Romain, Guyot. We belong to Universidad Autónoma de Manizales, and we are part of the Bioinformatics and Artificial Intelligence team. The Hemileia vastatrix fungus produces the phytopathogen that has most affected the coffee growing sector in South American countries, called coffee rust, destroying up to 30% of annual productions. Likewise, Hemileia vastatrix has one of the largest genomes among the fungi, being approximately 800 megabases. In previous studies, it has been visualized that 80% of this genome is made up of transposable elements, which are mobile sequences in the DNA that cause changes in the genome. Specifically, the order of LTR retrotransposons present a significant contribution in the genome of organisms such as plants and fungi. Despite the fact that there are two available genomes of Hemileia vastatrix, it has not delved into specific elements of the genome such as transposable elements. That is why in this project we decide to make an annotation of Hemileia vastatrix, noting the impact of the lineages of LTR retrotransposons in the genome size. For the methods, we first have a data recollection stage, in which the two current genomes of coffee roast, HB33 and HB-CAT, were investigated and obtained. Subsequently, a database of transposable elements was built. For this stage, the softwares Repeat Modeler, Repeat de Novo, LTR Struct, ETA, LTR Finder were used for identification. Impactor was used for classification, Repeat Masker for the annotation, Bowtie2 for mapping, and the CD-HIT software was used for the clustering process. Then we proceed to make the annotation of the transposable elements. At this stage, we have also an analysis of the insertion time, an estimate of the genome size. Finally, the results are discussed, in which the analysis of the contribution of transposable elements in the genome size of the species is carried out. We continue with the results. Several families and lineages not previously reported in the literature were identified. In the phylogenetic tree, we observed the distribution of the identified transposable elements. We can also note that two of the new lineages, Cowie and Soroa, are the ones with the most impact in genome size. This can also be observed in Figure 2, where it can be seen that both Soroa and Cowie, noting that Andes, Betania, Peregrino, and Amani belong to this lineage, are the ones that present the greatest contribution in the genome size, with a 37%. All of these lineages belong to LTR order and have a total contribution of the genome about 50%. We can also appreciate that line order provides less than 2% of all genome size, and that the DNA transposons, also called class 2, provide less than 5% of all genome size. Likewise, in the insertion time estimation graph, it is observed that Betania family belonging to Cowie is the one that has been inserted most recently, suggesting that it could have been activated recently. Finally, all of this information represents a valuable resource to understand the role of transposable elements in the evolution and pathogenicity of Hemileia vastatrix, being a significant contribution to future studies of this phytopathogen. Also, this helps to have a good approximation to understand why this genome is large, and for future studies we can think of compare different genome of other fungus species and make a comparison among them.